This video lecture is over Chapter 26. We'll cover reproduction and embryonic development. Let's begin with sex determination and differentiation. In humans, each one of our cells contains 46 chromosomes. Those 46 chromosomes are made up of 22 pairs of what we call homologous autosomes. And homologous means that each one of those chromosomes contains the exact same genes. So 22 homologous autosomes. And then we have a pair, so our 23rd chromosome is a sex chromosome, and we have a pair of these. If you're female, you have two X's. If you're male, you have an X and a Y. In the ovaries and testes, those cells undergo meiosis to create sperm and eggs that are haploid, meaning they only have a single copy of each of those 23 chromosome pairs. So we'll have 22 autosomes and one sex chromosome in each of our gamete cells. Sex is determined by whichever gamete chromosome the sperm carries. So if the sperm has a Y, then the fetus will develop into a male. If the sperm has an X, the fetus will develop into a female. And that's because the Y chromosome contains a gene called the SRY gene, and it codes for a protein called the testes determining factor that'll cause the differentiation of the internal reproductive organs that become the testes in a male. If we lack the Y, then those internal organs will develop into the ovaries in the uterus. Before we talk about that differentiation, let's clarify a few things when we talk about sexual characteristics. So we can divide sexual characteristics into what we call primary sex characteristics and secondary sex characteristics. Primary sex characteristics are the internal organs and external genitalia. So in a male, that would be the testes and the penis. In the female, that would be the ovaries, the uterus, and the vagina. These genitalia are going to form from what we call bipotential tissues that could become either or. The secondary sexual characteristics of an individual are the traits that we often associate and distinguish males from females. Those would be things like body shape, the body hair, facial hair, muscular development, lowering of voice, the libido, mammary tissue, like breasts in females. And secondary characteristics oftentimes can be enhanced through hormone and steroid production. So let's look at this cascade of events that occurs when we have a Y chromosome present in a fetus. As the fetus develops, the SRY region of the gene gets turned on, and we start producing testes determining factors. These proteins initiate the testes to start developing. And we start developing cells in the testes that are interstitial cells and Sertoli cells. So interstitial cells start producing testosterone. And when we have testosterone, that then binds to receptors on the Wolferian ducts. I'll show you a picture of this on the next slide. And the Wolferian ducts then start to form into the epididymis, the vas deferens, the seminal vesicle, and we start getting our primary sexual characteristics of a male development. At the same time, additional cells are being stimulated by the TDF proteins. Those are Sertoli cells, and they produce a hormone called the anti-Mullerian hormone. That hormone binds to receptors in the Mullerian duct, which the Mullerian duct can become the ovaries and the uterus and the fallopian tubes, and this causes those to regress and to atrophy. So if we do not have the SRY, if we don't contain a Y chromosome and we don't have the SRY gene, we will never produce these, so the interstitial cells and the Sertoli cells will never start to form, and instead the ovaries will begin to differentiate, and the Wolfarian duct will regress. So let's look at this. I just went through that process. This is another image that kind of goes through that. Again, looking at around a 6-week-old embryo, they are what we call bipotential, in that if we're looking at the internal structures, we have both the Wolfarian and the Mullerian ducts. And depending on whether we have that SRY, if we have that SRY, that around 10 weeks, we're going to start producing testosterone because of the production of those cells in the testes, and that's going to cause the Wolfarian ducts to develop. So we see it right here. We start developing the testes. We start producing the anti-Mullerian hormone, and the Mullerian ducts will atrophy and regress and disappear. These will turn into our testes, our epididymis, vas deferens, etc. And we'll have, by the time of birth, we'll have the internal primary sexual characteristics developing. Likewise, if we don't have this, right, then instead at 10 weeks, what we'll see is that the Mullerian duct starts to develop, and the Wolfarian duct actually starts to degenerate because there isn't enough testosterone being produced. And so we'll develop the ovary, the fallopian tube, and the uterus at birth. Likewise, if we look at the external genitalia, we'll see at 6 weeks, again, we have this bipotential stage where we have the genital tubercle and the labioscrotal swelling, urethral grooves, and depending on those sexual hormones that are being produced, testosterone in the male and the androgens in the female of estrogen and progesterone, we'll see the differentiation and development of the external genitalia. So in the male, we'll start developing the glands of the penis, and the labioscrotal swelling will fuse and will develop the scrotum, whereas in the female, we will develop the clitoris, and the labioscrotal swelling will divide into our labia majoris, minus, and we'll have the external genitalia of the mons and the labia and the clitoris forming in the female. So both of these are coming from the same bipotential tissues, and it's directed by testosterone and estrogen being produced. Our brain plays a direct role in reproduction. We produce what we call gonadotropin releasing hormones from the hypothalamus. And these hormones are going to play a role in the development and functioning of the different cells found in the ovaries and the testes. The two main gonadotropin hormones that then get released from the anterior pituitary. So gonadotropin releasing hormone stimulates the release of the follicle stimulating hormone, what we call FSH, and that is the primary hormone that deals with gametogenesis, so the formation of gametes. And then we also produce the luteinizing hormone, or LH, which is largely responsible for the production of our steroid androgen, or steroid hormones, either estrogen or testosterone, in the tissues, and progesterone as well. These sex steroids that get produced are very closely related to one another. We have enzymes like aromatase that can convert these precursors, what we call androgens, into our estrogens. And both sexes, whether you're male or female, you're going to produce both androgens and the estrogen. Okay, the dominant sex hormones or androgens in male is going to be testosterone. That's going to be the more dominant. In females, it's going to be estrogen. And we produce estrogen from the estradiol or estrone and found in the ovaries, and the progestins and other androgens will be turned into testosterone and progesterone. So if we look at how this gets triggered, again, we'll see that we have the gonadotropin releasing hormones. It gets released by the hypothalamus. That stimulates the anterior pituitary to release the luteinizing hormone. And the follicle stimulating hormone. And then we'll see that that'll lead to gamete production. In females, the luteinizing hormone also plays a little role in gamete production. And the luteinizing hormone will produce or stimulate our endocrine cells to create our sex steroids, our testosterone and estrogen. And there's feedback loops that play a role in these as well. So let's start with male reproduction. A little bit of anatomy. The external genitalia of the male is going to be the penis, right, and the scrotum. Inside, the internal organs are then going to be the testes. We have from the testes, we'll have the epididymis. Then we have the vas deferens, and we have accessory glands associated with the male reproductive. So we have the prostate gland and the bulbourethra gland, sometimes called the Cowper's gland, and the seminal vesicles. It'll play a role in producing the seminal fluids that are needed for sperm production and longevity. Okay, so sperm themselves get produced in the seminiferous tubules that are found in the testes, and this is where we have all the Sertoli cells that support those gamete production. So we have the Sertoli cells found in the testes. They're being stimulated by the follicle stimulating hormone, and they are providing the proper proteins and productions that's necessary for developing sperm to mature. They also produce hormone called Inhibiting, which has a negative feedback loop on FSH. And we'll talk more about Inhibiting with female maturation in a few slides. The other cells that we have in the testes, so Sertoli, and we have interstitial cells. So interstitial cells are the ones that are being triggered by luteinizing hormone to produce the testosterone, and that androgen is important for hormonal balance and functioning in the body in general. A few notes on sperm themselves. So the sperms are the gamete that we produce. There's not much to a sperm in the sense that it's mostly a nucleus. There's not a lot of cytoplasm left in this cell, and all of the mitochondria have been pushed into what we call the midpiece, and they're utilized for whipping the flagella and the microtubule apparatus that allows for movement. At the head of the sperm, we have what's called the acrosome that contains enzymes that will be utilized in fertilization. In addition to sperm that are being produced in the testes, we also produce in a number of our accessory glands semen, which is a fluid secretion that's really it's necessary for sperm development and longevity. So here's some of the different components that we produce in the male reproductive tract. So sperm, its function as a gamete, it gets produced in the seminiferous tubules, and then we start producing all of these fluids that make up the semen. So mucus is produced as a lubricant. That's what's produced in the Cowper's gland and the bulbourethra gland. We add water to that mucus. Most of our accessory glands are going to provide that water. It's important that we put buffers into the semen. The purpose of the buffer is to help neutralize the acidic pH that's found in the vagina that would normally destroy sperm, and that's produced by the prostate gland and again the bulbourethra. And then there's nutrients that are getting produced in different parts of the reproductive tract. So we produce fructose, which is going to provide a energy source for the cell. citric acid, vitamin C. These are all important to ensure that the sperm has the energy for fertilization. And those get produced in the seminal vesicle, the prostate, the epididymis. And then there's a few other things that are really important for sperm functioning. So there's enzymes that get produced in the seminal vesicles that help the sperm move through mucus found in the female reproductive tract. We know that there's zinc in semen, and we know that it's important for fertility, but we don't actually know how it necessarily functions. And then finally, we have a bunch of what we call prostaglandins, which are hormones that are produced that play a role in smooth muscle contraction and aid in sperm being transported through the vagina, uterus, and the fallopian tubes because they cause smooth muscle contraction. One last point I want to make about male reproductive anatomy and physiology. So the formation of gametes in males is what we call spermatogenesis. And spermatogenesis will begin around puberty when testosterone starts being produced in large amounts. It will continue throughout the duration of the lifespan of that individual. And so we'll have a continual production of spermatogenesis that lasts pretty much for as long as that male is alive. So we will, on a daily basis, be making tens of thousands of sperm which brings us to female reproduction. So, quick overview, the external genitalia in the female would be the vulva, the labia majora, labia minora, the clitoris, right? And then the internal organs are going to be the vagina, cervix, the uterus, fallopian tubes, and then the ovaries themselves. Ovaries have two main functions. First function is going to be oogenesis, so the production of ova, the production of eggs. The second function is going to be the production and secretion of our sex steroid hormones, which are progesterone and estrogen. In the ovary, we have a number of follicles that are developing. These are cells that contain those gamete cells, and we'll go through these in just a second. So our ovarian follicles are what we could call the functional unit of the ovary, right? This is where we will produce those ova cells that can potentially become a fetus. They provide nutrients to our oocytes during development. So the follicle isn't just the oocyte, it's the cells associated with it. And those associated cells provide that nutrients to develop the egg. They help release the egg at ovulation. They produce the sex steroids that are going to prepare the uterus and the fallopian tubes to help fertilize and promote fertilization of sperm coming in. They're going to prepare the uterine lining for implantation of the fetus if it becomes fertilized, and they help maintain the sex steroids during early fetal development while we're waiting for the placenta to develop. There are 5 developmental stages of a follicle. The first stage is what we call the primordial follicles. These are where the cells are just very small. We have a single layer of cells that surround the oocyte. So you can see it right here. There's the oocyte, and there's maybe a few cells that surround it. We then move into what we call the primary follicle, where we have more of these what we call cuboidal, and they're granulosa cells. So you have granulosa cells in the primary follicle. We can then move into a secondary follicle, where we start seeing this formation, what we call the antum, where we will start having fluid coalesce there. We have granulosa cells, and we also start to see some of our theca cells. And these secondary follicles can then become what we call a primary follicle or a Graafian follicle. And those Graafian follicles, you still see the oocyte in the center. There's the antum, all the granulosa cells. These are what will actually have the where ovulation will take, and it'll rupture. We'll release the ovum out. After we've ovulated and released the ovum, this follicle will then turn into an endocrine gland that we call the corpus luteum, and it'll slowly degenerate from there. So let's go through oogenesis and what this looks like. So oogenesis begins in females, unlike males where it begins at puberty. In females, this begins during embryonic development. So during the first 20 to 24 weeks of embryonic development, the female fetus will produce around 7 million eggs. Okay, they've started their first mitotic division. Okay, so mitosis has occurred, and we have these primordial follicles, but we haven't yet done meiosis. We just are beginning to start meiosis. So all of our oogonia, they enter prophase 1 and become oocytes, and that occurs somewhere between when we're gestating to about six months postpartum. So after the fetus has been born, the baby's been born, and then they stay suspended in prophase 1, and they are locked in suspended animation until they get triggered to develop further. And what we see is this attrition, this degeneration that occurs. So where we started with maybe 7,000 oogonia at birth, 7 million oogonia at birth, that's dropped to maybe 2 million. By the time we hit puberty, maybe there's 400,000. And when we get to menopause, there's maybe only a handful that actually remain. And unlike sperm cells, where we're continually producing new spermatogonia, new oogonia are not produced in the ovary. So you start with 7 million and you lose them from there. Okay. During childhood, the ovary is inactive and everything is arrested and kind of staying in that primordial follicle stage. And then at some point, when we start puberty, and we start producing the sex steroid hormones, and the anterior pituitary starts producing follicular stimulating hormone and luteinizing hormone, and the body starts increasing its production of estrogen and progesterone. We start in a few cells development. So not all of the cells will go into development, just a few will start developing. And they'll develop into a primary follicle. So we get this primary follicle, and it starts growing, and we see the granulosa cells start to proliferate around the outside of the follicle. And the purpose of the granulosa cell is to nourish the oocyte with nutrients, with hormones, and they secrete fluid into the antum. These are very similar to the Sertoli cells that we see in the testes. Now, the theca cells, they're going to start developing, and those are our other cells, and they're largely important in producing the sex steroid. So they would be analogous to the interstitial cell in the male reproductive tracts. So at puberty, this starts to occur. Each month we will go through this cycle. So each month we'll have a few primordial follicles become primary follicles, and a handful of those primordial follicles will actually enter what we call the second stage, where they'll become a Graafian follicle. And this takes time. This is constantly occurring. It's kind of in a monthly cycle, but to go from a primary to a Graafian, it's going to take something like 80 days. Okay, And so this is constantly kind of occurring from puberty on, that we're having this maturation, this development. And you'll notice by the time we get to a gray fin follicle, we have completed meiosis 1. So we've gone through one division of meiosis. We're now haploid, but we still have 2 copies of all those haploid, 23 chromosomes. The other cell that gets produced, so we don't produce 2 gametes. The second gamete is what we call a bar body, and all the cytoplasm is pretty much retained in the original cell, and so we're only developing 1 egg. Whereas in spermatogenesis, for each gamete that we begin with, or for each beginning cell, we end up with 4 sperm cells. In females, we'll start with a single precursor, so a single primordial follicle, and we'll only end up with 1 ova cell, 1 egg cell. This puts us into the third stage, in which now we may have a handful of these gray fin follicles, but only one will become what we call the dominant follicle, in which it really starts rapidly developing. This development starts occurring now in the matter of days. So in about 5 to 7 days, it really starts growing. It dominates above all the others, and it actually starts producing some compounds that inhibits other follicles from further developing. And then in a very short period of time, in a matter of hours, this will get triggered to ovulate and will rupture the dominant follicle, and ovulation will occur. And you'll see at the same time what's going on with meiosis. We're starting meiosis 2, but meiosis 2 actually doesn't complete until we have fertilization of the sperm. So we release this oocyte, and after it's been fertilized, we will complete meiosis 2 of that egg, and will now start developing it into the fetus. The sperm's DNA will mix now with the egg's DNA, and we'll start fetal development. After ovulation, I mentioned that now that follicle will turn into what we call the corpus luteum. This is mostly granulosal cells, and there's a handful of theca cells associated with it too, and it becomes an endocrine gland where all it's producing now is steroid hormones, primarily progesterone, which is going to help with the now fertilized zygote implanting into the uterus. And we'll go over that. So this is just another figure taken from your textbook. It's figure 26.10, and it kind of goes through. Don't worry about all these different layers, maybe just granulosa cells, theca cells, but you can kind of see what's going on during these different stages. Now we're going to talk about the hormone regulation of this development that's happening during oogenesis. So as I said, during childhood, the ovaries are growing, but they're not really producing sex hormones. So there's a very low amount of estrogen that's being produced. And this low level of estrogen, it inhibits the hypothalamus from producing that gonadotropin releasing hormone. So we're kind of in this stasis. And as we near puberty, the level of estrogen sensitivity that occurs in the hypothalamus starts to change. Where the hypothalamus, really wasn't all that sensitive to low levels of estrogen, now we start to get a lot more receptors, and it becomes less sensitive to it. Okay, so it isn't being affected as much by just the amount that we have that low continuous amount, and so at this point we start producing this gonadotropin releasing hormone. And we call it pulsatile because we just have these ebbs and waves of producing this hormone. As we produce the gonadotropin releasing hormone, that stimulates the anterior pituitary to then create and secrete the follicle stimulating hormone and the luteinizing hormone. So as a girl nears puberty, this could take around like 4 years where this is is starting to kind of come online, in which we're starting to become really regular. And at some point, that girl will have her first menarche, which is their first menstruation. And after that, again, it's going to take probably 3 years for her ovarian and uterine cycles to be what we'd call more regular and ovulatory, to where there's a regular ovulation occurring. Don't worry too much about this information. I put these slides on here to give some additional information about FSH and luteinizing hormone. Really what I want you to understand about FSH is, first, it's a glycoprotein, and only granulosa cells have FSH receptors. So theca cells don't respond to FSH. What FSH does is it stimulates those primary follicle granulosa cells to grow. And as they grow, those granulosa cells start producing estradiol, or estrogen. Luteinizing hormone, very similar to FSH, but its role is to initiate ovulation. So you can remember luteinizing hormone for corpus luteum, that comes after ovulation, and it's going to bind to receptors and stimulate the formation of the corpus luteum. In women, there is what we call the menstrual cycle, in which we go through this monthly cycle of hormones that directs the ovulation of oocytes. In general, females release gametes around every 28 days. It can differ. It can be short as maybe 24 days. It can be 35 days. That's considered 24 to 35 is normal. It could be much longer in some people if it's abnormal. But this would compose what we call the menstrual cycle. There's 2 cycles that make up menstruation. There's the ovarian cycle, so this is what's going on in the ovaries, and there's the uterine cycle of what is going on in the uterus. In the ovarian cycle, in the ovaries, there's 3 stages. There's what we call the follicular stage, in which we have follicle growth. There's ovulation, and there's the luteal phase. In the uterine cycle, it begins with menses, and then we move into what we call the Plarificative Phase and the Secretory Phase. So I'm going to go through these slides. I have a couple different slides that show the hormone interaction, and then kind of a slide at the end that summarizes it all up. But it's really important that you guys are able to kind of go through this and follow what's going on. So this is the ovarian cycle. Again, 3 stages: follicular stage, ovulation, and luteal stage. And what we begin with is, again, that gonadotropin releasing hormone stimulates the anterior pituitary to release follicular stimulating hormone and luteinizing hormone. And as that stimulates the primary follicle, it starts to grow. Now, why are some follicles targeted? I said not all follicles grow. Only a handful each month are being triggered and starting to develop, and it takes 80 days to develop. So why do some start to get triggered and others? Well, we think that this has to do with the presence of receptors in the membranes of these cells, and so some of these, at some points, there's processes that trigger more receptors to be placed into some of these follicles, and that's why they get triggered to start. Okay. So the FSH is binding to the granulosa cells, the luteinizing hormone is binding to receptors on the theca cells, and as that follicle enlarges, the theca cells start to produce androgens. Okay, they start producing estradiol, and estradiol gets converted in the granulosa cell into estrogen. So what we see is, as these start to develop, estrogen levels or estradiol levels in our blood starts to increase. And that has a negative feedback loop on the anterior pituitary. So this negative feedback loop says, let's stop releasing FSH and luteinizing hormone, which is important because we don't want additional follicles to maybe start developing. Okay, so we start inhibiting this release. Now, we're still producing FSH and LH. We're just not secreting it. Additionally, the granulosa cells produce a compound called inhibin B. And this has a strong negative feedback loop on the anterior pituitary and specifically on the follicular stimulating hormone itself, to where now follicular stimulating hormone isn't even being produced. So we're still producing luteinizing hormone, but we're not releasing it, and we've stopped really producing follicular stimulating hormone. The cell keeps developing, but at some point now it's become a mature follicle, and that dominant follicle, when it starts to rapidly develop, we start producing a lot of estrogen. And when estrogen levels get high, we have this critical limit that gets hit. And it's at around 200 picograms per milliliter, in which when we go above that, now instead of having that negative feedback loop, we turn into a positive feedback loop. That triggers the hypothalamus to really start, it triggers the anterior pituitary to really start binding and upregulating its gonadotropin releasing hormone receptors. And so it binds a lot, and it basically says, hey, start secreting and producing FSH and LH. Meanwhile, we've been storing all this LH in the anterior pituitary, so what we have is a ovulatory surge of the luteinizing hormone and the FSH. So a bunch of luteinizing hormone gets released. That triggers that mature follicle to rupture, and we have ovulation. And after ovulation, then the corpus luteum is being supported by the luteinizing hormone that was released, and that triggers it to produce estrogen and progesterone, which has a negative feedback loop, again, on the hypothalamus and the anterior pituitary, to where they don't release FSH and luteinizing hormone anymore. So I went through all that. I didn't click through the slides, but you can kind of go back and through and look at all this. But what we see is this cycle kind of going. And I'm going to jump ahead a little bit. I'll come back to it. But this right here shows what's kind of going on during the follicular stave, ovulation, and the luteal phase of the ovarian cycle. So again, estrogen is kind of low. It starts to increase a little bit. We have, FSH and LH that are, mid, and then they start being inhibited by our estrogen production and inhibin B. We have that trigger in which we have the surge. Estrogen rises. That triggers, a bunch of luteinizing hormone to be released. We don't have as much FSH being released. And then when we get into the luteal phase, you'll notice what we primarily are producing, what the corpus luteum is producing, is progesterone. So we still produce estrogen, but not nearly, that's not the main androgen. It's now the progesterone. And you can see all the feedback loops and what's producing from this nice little figure right here. All while the ovarian cycle is going on, we have processes that are being affected by these androgens in the uterus as well. So I said the first stage of the uterine cycle is menses, or menstruation. So from day 1 to 5, the uterus is shedding its outer layer of epithelial cells. So we call it the stratum functionalis. That's when during this stage it's associated with blood, bleeding, blood loss, tissue loss that's passing out through the vagina in what we call the menstrual flow. Okay. At this point, the ovarian hormones, they're at their lowest level. So estrogen and progesterone are really, really low. We start seeing the follicular stimulating hormone and luteinizing hormone beginning to rise. And at about the 5th day, that's when our growing follicles start producing estrogen. And so we see an increase in estrogen. And this is when we enter into the stage 2, what we call that plarificative phase. This phase can last anywhere from 6 to 14 days, depending on how quickly it happens. And during this time, what's happening during the plarificative phase, that estrogen is causing that stratum functionalis to really start generating and growing. So the stratum basialis generates a new stratum functionalis, we start getting the arteries and glands start increasing in size, and we start getting progesterone receptors that are being placed in developing in this endometrial cells that we find in the uterus. Also during this time, the estrogen is causing what we call that thick, sticky cervical mucus that acts as a barrier between the vagina and the uterus. Normally it's thick, it's starting to thin out and becoming crystalline, and we see that, right? So it's pretty fairly thick, and then right here we see prior to ovulation, what's occurring is you start to get these crystalline forms. This allows sperm to kind of move through these channels and move past the cervix. Okay, once we ovulate, that release of luteinizing hormone, right, very quick, that marks the end of the proliferative phase, and we now move into the secretory phase. And in the secretory phase, this is a 14-day cycle. So it will always last 14 days. Just like in the ovarian cycle, the luteal phase, it will last 14 days. Menses can differ, number of days it lasts can differ. Plarificative phase, the number of days that can last can differ. But the secretory phase and that corresponds with the luteal phase will always be 14 days. So you can back calculate when ovulation occurred. And we'll do that in just a second. So during the secretory phase, the primary androgen is progesterone that's being produced. And what that causes is all of this rapid growth of the stratum functionalis, and we start having these spiral arteries elongate. We start bringing glycogen is being released into this tissue, and those are nutrients that are needed to sustain the embryo before it implants. So we start releasing glycogen into the uterine cavity. Likewise, that progesterone causes the mucus on the cervix to create a plug to prevent additional sperm from entry. We start having that as that progesterone rises and that inhibin rises, we stop producing that luteinizing hormone. So we'll see those kind of just drop down. No more luteinizing hormone, no more follicular stimulating hormone. And if fertilization doesn't occur, then the corpus luteum that's producing all that progesterone and estrogen, it won't be able to sustain itself for more than 14 days. And what we'll see is that there's a drop-off in the progesterone and estrogen, and when that starts going down, that triggers these spiral arteries to start kinking and spasming, and as they kink and spasm, they shut off oxygen, or they don't allow oxygen to flow into this tissue, and so we start having tissues that die, they rupture, they start self-digesting themselves, and that triggers the onset of menses, and we start to cycle over again. So again, here's that thing. We're putting everything together now. So on top, I love this figure. Here we can see the ovarian cycle. Here we see the uterine cycle. So ovarian cycle starts with follicular phase. Then we have ovulation and luteal phase. The uterine cycle is menses, plarificative, and then secretory. And this half, the second half, will always last 14 days. So the luteal phase, the secretory phase is 14. So if an individual has a cycle that is typically 30 days long, you can calculate on what day they ovulated by the onset of their menses. So let's just say on the 1st of March, this woman started her menses. And we know that she has a 30-day cycle. Then that would mean that we would expect her to ovulate on 30 minus 14. So that would be on day 16, we would expect that ovulation to occur. In a 28-day cycle, it's 28 minus 14 is on day 14. So make sure that you can walk through this. I know this is a lot of information that you can understand these negative, positive feedback loops that kind of occur in this regulatory stuff. A lot of the questions from your study guide kind of walk you through this of saying, hey, outline this, how does this occur, how does that occur? There'll be a lot of focus on understanding what's going on with this cycle. Understanding this cycle is really kind of the key to understanding a lot of biology that surrounds contraceptives and how we use contraceptives. So we can classify contraceptives in a number of different ways. There's three broad types. We have what we call barrier methods, in which you try to prevent entry of sperm into the reproductive tracts. Those are going to include spermicides and diaphragms and condoms. Even abstinence technically would be a barrier method, right? And then we have hormonal treatments, which would be like our oral contraceptives, our contraceptive hormonal implant. And then we also have what we call methods that seek to kind of reduce implantation of a zygote. Or at least that's kind of how I used to classify it. Now we tend to think of things as like intrauterine devices that a lot of times kind of make it more difficult for something to implant. And let's talk about a few of these and how they work. So let's just take out barrier. We don't need to talk about those. I think those are self-explanatory. But if we look at hormonal treatments, if a woman is taking oral contraceptives, and what we're trying to prevent is ovulation. We're trying to prevent the oocyte from being released. If it's not released, there's not any ability for fertilization to occur. And so how can we maintain that? Well, once you understand these cycles, you recognize that ovulation is triggered by the luteinizing hormone. So if we never have this surge in estrogen, right, that causes that surge in the luteinizing hormone, we won't have ovulation. So a typical woman that's taking birth control pills is taking hormones, probably progesterone and maybe some estrogen as well. And they're ensuring that they keep kind of a low level of estrogen and progesterone that doesn't have, that prevents this buildup of ovulation. That still means that you're going to have some follicular development. It just means that those eggs are never going to ovulate. Now, at the end of a contraceptive pack, typically there will be a period in which a woman takes, like five to 7 pills that are just sugar pills. There are no hormones in them. And the purpose of that is to trigger menses, right? So we still have plarificative phases. We still have secretory phases. They may not be as full because we might not be having as much progesterone or not. But as we start to get further in that cycle, we start increasing the progesterone more and more and more, and then we cut it off. Those sugar pills basically force us into this, where we drop to very low levels of progesterone, and that triggers the menses. Let's talk about intrauterine devices. So here you're having a device implanted into the uterus, and it serves a couple of different functions. First off, most of them have a copper associated with the device. Copper makes it difficult for sperm to maintain motility, so they can't swim. It kills the sperm off. So on one hand, they're preventing sperm from reaching fertilization. Some of these intrauterine devices also have a hormonal component to it that seeks to prevent, again, the luteinizing surge. And so there's a progesterone that's kind of produced to prevent that from happening. And then some of them also then will have a mechanism that doesn't allow, if an egg were to be released and if it were to be fertilized and travel down, it wouldn't allow for implantation to occur and that initial cellular growth and development. So that would be an intrauterine device. Now, not all work that way. We also then have like contraceptives, birth control that are like the morning after pill, which sometimes is discussed in controversial terms, but really there's nothing controversial about it. All we're doing again with morning after pill is a high dose of progesterone, and the purpose of that is kind of threefold. One is to prevent ovulation. So if you haven't already ovulated, it seeks to prevent that. Two, that high level of progesterone creates a mucus plug that would prevent sperm from entering in. So even, if perhaps you had already ovulated, but if you were to take that, you'd prevent sperm from being able to move into the fallopian tubes past the cervix. And then there's, to a far, far lesser extent, there's some evidence that shows that it may make it a little bit more difficult for implantation to occur, but those aren't the primary mechanisms by which that pill would work, right? So it's mostly to prevent ovulation and to prevent a blockage, a barrier method to sperm be able to move past. So thinking about contraceptives and how these things work and how we can manipulate these hormonal fluxes in the body is really important for us to understand so that we can make decisions about family planning. Let's move on to fertilization now. So sperm mature once they are actually in the reproductive female tract. What occurs is the sperm will then go through a process called capacitation, in which it starts producing hormones or producing proteins in the acrosome, and it allows it to kind of decode itself and become able to rapidly swim and move. Without it, the sperm isn't going to actually move very well without capacitation. This is one of the barriers to in vitro fertilization. If you're going to take sperm from an individual and then try to artificially inseminate somebody, or in a petri dish fertilize an embryo and then be implanted, you have to go through this process of capacitation. And so we've sought to try to do that by putting them in saline solutions that mimic some of the pH and secretions found in the reproductive tract to make them active. Once they've gone through capacitation, a sperm is viable for about 5 to 6 days in the reproductive tract. They will move slowly through the reproductive tract. They will attach themselves in the fallopian tubes and hope to fertilize an egg. When an egg comes by, the sperm are attracted to that egg by chemical cues. The acrosome will then penetrate this glycoprotein coat. And when it does that, the sperm then injects some enzymes into the glycoprotein coat that breaks it down and allows it to maneuver to where it actually can get its head into the egg. Once the head is in there, it's going to release the nucleus of the sperm into that. And additionally, that sperm also releases some proteins that kill other sperm around. And at this point, the egg will complete its second mitotic division. It'll create the second polar body. And then that second polar body, now it's already was haploid, but now it only has one set of chromosomes, and that those will combine with the male's sperm's chromosomes, and the zygote will begin to form. This takes us to the steps of implantation. So we had ovulation. At some point after ovulation, there was fertilization, and then it takes about 4 to 5 days for the zygote to move out of the fallopian tube and into the uterus to start implanting. During that time, the zygote is going to divide like 50 times to where now we have what's called a blastocyst, and we're starting to form different layers in these cells. We have the blastocyst. There's an inner cell mass that's going to turn into the fetus, but we start developing the chorion and the amnion tissue layers. Once we get in the uterus, this is about 7 days after fertilization, we start to secrete, the blastocyst will secrete enzymes that allow it to invade into the endometrial cells of the uterus, and it kind of implants itself in there. And in that outer layer, said forms the chorion, the amnion, the allantois, and the yolk sac. In humans, our yolk sac doesn't last very long. What I want you to know is that it's the chorion that starts to become the placenta. It forms the placenta. It creates what we call these chorionic villi, in which the blood vessels that are forming in the umbilical cord and in the fetus start to migrate through, and we create this blood barrier between the maternal blood and the chorionic villi, where we have the blood exchange. At this point, we now are developing the placenta, and the placenta will start to secrete its own androgens. And the placental hormones that get secreted, there's 4 that I want you guys to know. The first is the human chorionic gonadotropin, or HCG. The purpose of this is to ensure that the corpus luteum keeps producing progesterone. So if there's not fertilization, after about 7 days, the corpus luteum is going to just die on its own. However, if we have fertilization and we start to form the placenta, we start producing this human chorionic gonadotropin, and it binds to the luteinizing hormone receptors on the corpus luteum and facilitates the continued production of progesterone. And we need progesterone to ensure that the uterine lining stays healthy. That'll happen for about 7 weeks. So the corpus luteum will produce progesterone for 7 weeks, at which time the placenta is now large enough and capable to produce its own progesterone to ensure that the uterus is nourishing for the fetus. If we look at hormone release, what we see is that HCG, it peaks at about 3 months, right? So at 3 months, it starts to diminish and we don't get much more of it. This is the hormone that early pregnancy tests utilize to detect if a woman is pregnant. Because again, shortly after fertilization, we'll start producing small amounts of this HCG. The next one is similar. It's human chorionic somatomammatropin, or HCS. This is a compound, a hormone that resembles growth hormone and prolactin in women. And it plays a small role in the development of lactation. It plays a larger role in altering the mother's glucose and fatty acid metabolism to ensure that it can alter its metabolism to support fetal growth. That's HCS. The placenta will then produce estrogen, which contributes to at this point of developing the milk secreting ducts in the breasts. It'll produce progesterone. Like we said, it maintains the endometrium and it suppresses uterine contractions. So when we don't have a lot of progesterone, we start to get those uterine contractions. Again, that's what triggers, menses. And lastly, it'll produce its own inhibin, like inhibin B, that prevents additional follicular stimulating hormone and luteinizing hormone from being released so that we don't have additional ovulatory events. So all these hormones are being produced by our placenta. The baby is now dividing and growing, and there's a whole course that you can take on human embryonic development that goes into all the processes that are happening at this point. What I want to just cover is what happens at the end for the physiology of now what we call parturition, which is childbirth. So around the 38th to 40th week of gestation, we start having rhythmic contractions in the uterus. That's what we call labor. And we don't fully understand what triggers labor. What we do know is that progesterone can play a role in it, that oxytocin can play a role in it, but both of those generally happen once labor already starts. There's a couple other hypotheses that are being studied, but it's still largely kind of unknown. I don't want to say completely unknown, but what actually triggers that onset of labor. What we do know is that once a labor starts, there's a lot of positive feedback loops that progress labor and cause it to advance. So we know cervical dilation. So the cervix has to soften. We call it ripening, and it has to dilate in order to allow the head of the infant to move out. And what triggers that is going to be the hormone Prolactin that helps the cervix soften. And as the baby's head pushes on the cervix, we're going to have release of oxytocin. And so oxytocin is going to get released. That causes the uterine wall to produce more prostaglandins. Those prostaglandins cause the uterus to contract. As it contracts, that's going to push the head of the baby harder on the cervix, which is going to cause more oxytocin. And we just have this positive feedback loop until the baby fully passes out of the birth canal and exits through the vagina and is delivered, and we have parturition. At that point, these cycles will stop. After parturition, the last physiological processes that go on in the woman's body is going to be lactation. So all of the mammary glands and the milk ducts were during the later stages of pregnancy developed through estrogen secretions. But once we start nearing the end of pregnancy, lactation starts to become triggered because the body usually is producing what we call prolactin inhibiting hormone, PIH. That PIH is being released and it stops the body from releasing prolactin. Prolactin is what causes the milk to be produced. And as we get towards the end of pregnancy, we start producing less of this. It gets weaker. And so we start having more, we have this decrease of it occurring. So more prolactin starts to increase. We start circulating more prolactin. And prolactin in combination with oxytocin leads to milk production. When we actually have the mechanism of letdown, where milk is actually pushed through and secreted, letdown is triggered by oxytocin and suckling. So when the baby suckles on the nipple, there's mechanoreceptors in that nipple that send a signal that further inhibit PIH. They also trigger the hypothalamus to trigger the anterior pituitary to release, the posterior pituitary to release our oxytocin, and that causes smooth muscle contraction in the mammary tissue, which leads to milk ejection. And that's